Good morning, student. We are in TYBSc Zoology Semester 1 Paper 2 Mammalian Histology class. We are discussing about elementary canal, histology of elementary canal, and that we have started with the previous two section of this lecture. We discussed about the gross histology of digestive tract. Then we have seen the detailed structure of esophagus, histology of esophagus, and some part of a stomach histology we have seen. Now we are in middle of stomach histology discussions. So this is a part 3 lecture of elementary canal. So this gastroesophageal junction is a location where esophagus connect with the stomach. Okay, so being a part of esophagus in this photograph, you will see that this uppermost portion having a stratified squamous epithelium in a mucosal layer, and this is exact point where the stomach started and esophagus ends. So at these junctions level you will find starting of different types of mucous epithelium, mucous epithelium. So here you will see a lot of gastric pits and folding of this mucosal layer, and that cell type of a mucosa also changes into the columnar type of epithelial tissues. Hmm. Here in another photograph again you will see all these stratified squamous epithelial portions is a junction point, and further they shows all that gastric pits and epithelial layer also changed. So this is the inner lumen portion and this is the outer portions of that muscularis mucosa and this esophageal glands. Here again this portion is a lumen and this is the outer portion of the submucosa, muscularis mucosa and everything. This is the inner lining. This is the inner lining. Here it is a stratified squamous epithelium. Here is the that columnar epithelial layer forming that gastric pits. Okay, again the same section of a gastroesophageal junctions, stomach and esophageal junction here. So this portion to the this side is all stomach portions and this is all esophageal portion. Okay, so you can distinguish this mucosal change here in between the two area. You can distinguish the changes in the mucosal area. Here again the same gastric mucosa and the pit. Now stomach portion they have shown under electron microscope. You will see all these rough surface indicate all opening of a gastric pit over the surface. This is so all these rough appearance indicate the opening of gastric pit. This muscularis mucosa, submucosa, muscularis, this externa, this serosa portion. Here cardiac and pyloric stomach comparatively, as you can see here comparatively structure of cardiac and pyloric stomach. Now this lower portion is the pyloric portions, and in our previous slide discussion we said that in a pyloric portions you will find more number of mucosal glands, whereas in these portions we find maximum all that secretory portions, that is gastric glands containing parietal cells, chief cells, zymogen secretions, mucus secretions. Okay, so basically both these epithelial layer are that columnar epithelial layer. Only modification is in a type of secretion in that columnar epithelial layer, which where they have these glands, and the glands Depending on their secretions and the locations, they refer as a cardiac gland if they are in a cardiac portion and pyloric gland in a pyloric portion. Okay, so here you can see the more details of these sections of a stomach, and you can see the gastric pits and the folding, that this is a fold upon that rugi, and the gastric pits here in the furrows of all these villi. You will see the here in this location, these are all gastric pits, and these portions are the major folds. So fundic glands, if that gastric glands are present in a fundic region, it is called as fundic glands. So gastric pits, opening portions, here is a neck portions, stemous portions, and opening portion is a mucoidal. So all these opening portion is lined with the mucoidal cells. Then these neck portions, below to the neck portions, there are all these enteroendocrine cells, chip cells, and parietal cells or axanthic cells. They are responsible for production of HCl. Chip cells or peptic cells are responsible for production of pepsinogens, which in presence of HCl convert into pepsin. Then these APUD cells are responsible for triggering the secretion of these peptic and axanthic cells. They are basically the endocrine cells responsible for secretion of the gastric juices in the stomach. And if you can compare this diagrammatic presentation here, so this portion is a gastric pit or the area lined with the mucoid cells, and in this gap here, if you trace any single gland, suppose here you will find three different coloration pattern. These dark color cells are there, light color cells are there. So these two different color indicate the two different types of cell here. That could be these parietal cells, and dark color could be of chip cells. And some are at the base. There must be some APUD cells. So on the basis of this changing color pattern in a single gastric pit or single gastric gland, you can separate that three type of secretory cells. Here again the fundic mucosa portion is there. These are the all rugi portions. These are the gastric pits, and here these all secretory, different type of secretory cells are there. Here again you can see the different color pattern of these cells. The dark color, light color, black color, and the pink color portions. So all that three different indicates the different types of cell. Basically, this they have used here the past stain, which gives color to the mucus. So all that mucoid cells are colored in a pink portions, which are not there at the below to that neck portion. So around the neck portions and in the opening of gastric pit, we see a lot of these uh, mucoid cells here. Then what exactly is a mucoid cell? These are the mucus secreting cells. Mucins is a secretion here which is poured over the surface, and it looks in the form of such a crumbed gelatinous material in a sections. Now in if you magnify this epithelial layer, mucoid layer of a stomach under microscope, you see this whitish color balloon shaped structure here on the surface of epithelium. All these white balloon structure indicates the accumulation of mucus in these mucoid cells, which is released over the surface. Okay, as this goes deep in a pit sections here, you will find such balloon structures are vanishing. So in these portions there is no mucoid cells are there. Majority all other these parietal cells, chip cells are there in these portions below to the neck of this gastric pit. Now, what exactly is parietal or axanthic cells are? They are secretory cells. The intracellular canaliculi are there in a single cells. You will find there is a highly branched and proliferating structures which is increase the secretory portions here. Lot of mitochondria in that, and they are pouring that secretion of HCl through this intracellular canaliculi. It is poured over the surface in that gland. So here you can see all these violet, these pink color portions are the axanthic or parietal cells. All these pink color structures indicate the parietal cells or axanthic cells here. This parietal cells ultrastructure is shown here. All these vacuoles and the mitochondria, their vacuoles are packed with the secretion of HCl, and all majority mitochondria are spread. Number of more mitochondria you see in a parietal cells. And chief cells and APUD cells. These argentophin cells or enterochromophene cells, or you can say the enteroendocrine cells, APUD cells. They are very few in number. Their role is in stimulation of secretion in the stomach gastric glands. Okay, now these chief cells are so called as a peptic cells. Here, this blue color structure indicates all that chief cells, and the gray color structure indicate the parietal cells in this actual photograph. So the role of chief cell is to produce the pepsinogen. Pepsi chief cells and parietal cell works is shown in this flow diagram, that chief cells produce renin and pepsinogens in presence of a secretion of parietal cell HCl. That pepsinogens convert into pepsin that is responsible for breakdown of protein into proteose and peptones, which is then absorbed and processed, passed into the liver and processed there for the protein synthesis further. Then also these parietal cells, they produce gastric intrinsic factor which facilitate the absorption of vitamin B12. Also, these APUD cells are responsible for some hormone-like secretions like gastrin, which stimulate the HCl and pepsin secretions. Then serotonin, when you eat that Cadbury or chocolate, this serotonin start producing in your stomach, and that cause stimuli that is responsible for the stimulation of smooth muscle contractions. You feel good. So okay, enteroglucagon, that increases the blood glucose level. So basically, these APUD cells are responsible they work as a enteroendocrine cells. The major secretory cells in stomachs are the chief cells and parietal cells. From here, we'll start with that small intestine. This small intestine is a tube about of 5 meter long, and it is divided into three parts as a duodenum, jejunum, and ileum. In sequence, the duodenum is a primary part, and it is about 25 centimeter long in human and 2 meters long in a as part is a jejunum, and the 3 meter long part is a ileum. Overall, that gut length is shorter in the living person than in the muscle tone conditions. So gross anatomy of a small intestine. Here you can see the pyloric sphincters and the bile duct and pancreatic duct and hepatopancreatic ampullae and ileoceliac walls. So if you see this diagram carefully, we can trace the all parts of this duodenum. So this after pyloric sphincters in a stomach, it start duodenum, and intestine start over here. It goes and kept in coiled form in abdominal cavity, which duodenum leads to jejunum and ileum here, and that then join with the large intestine at the portion where there is an appendix. So this runs from pyloric sphincters of a stomach and end into the ileoceliac wall. The bile duct and main pancreatic duct joins the duodenum at the hepatopancreatic ampulla. Now here at the curved portion only there is an opening where all these pancreatic secretions and bile secretions combine with the small intestine, where they pour their secretion into the small intestines. This ileum joins the large intestine at the ileoceliac wall, which is present just above the appendix. Basically, the small intestine divided into duodenum, jejunum, ileum. So, small intestine. They are the major site for absorption of nutrients, and its mucosa is made up of these microvilli, that is the epithelial layer, in the form of a villi and microvilli, that is a mucosal membrane. Then glands, here it is instead of glands they have used the word crypts. Crypts of lubricant in the lamina propria that open into the mucosa at the base of villi. In earlier histology of stomach and esophagus, we seen that epithelial layer in a esophagus is a stratified squamous epithelium. In case of a stomach, it is a columnar epithelial layer. But now in this small intestine, this epithelial layer, they have very fine microvilli over the surface of epithelial tissues. Okay. So there are three sections: duodenum, where you will find Brunner's glands present in a duodenum. In case of jejunum, long villi and increased global goblet cells are there. No Brunner glands or prayer patches you see in a jejunum. And in case of ileum, they have very short villi, many goblet cells, and lymphatic nodules in the submucosa called as a Peyer's patches. Now, histologically, all that histology is very much similar in all these three sections of a small intestine. So, to identify which portion, okay, we are discussing small intestine. A major site for absorption of nutrient and its mucosa is made for this. Villi and macrovilli are the major um, thing to note in a histology of GI tract. If you see a villi and macrovilli, that is intestine. Okay, then glands here are called as a crypt of lubricant, which is present same way like a gastric gland in a lamina propria. Then submucosa contain Meissner plexus, and this small intestine is divided into three different sections. Every section has a peculiar characteristic structures. In duodenum, you will find Brunner's glands. In jejunum, there is a long villi and increased goblet glands with um, in case of ileum, there is a short villi and many more goblet glands you will see, and large number of lymphatic nodules with Peyer's patches. So small intestine microscopic anatomy. Here you can see the structural modifications of the small intestines for increases that surface area. For increasing surface area, that small intestine is provided with a lot of micro that villi, finger-like projection extensions into the mucosa, and over to that there you will see on that surface again you will see the microvilli, that is tiny projections of absorptive mucosal cells. That extensions are there after of a plasma membrane. So many villi, on that villi again the microvilli are there. So overall the surface area is hugely increased. So here in a gross anatomy to the finer histological details are shown in this photograph of a small intestine microscopic detail. So we'll start with this first diagram here. Physically, these intestines are provided with the veins that carries blood to the hepatic portal vessels. So all that absorption take place from this intestine is poured through this blood vessel into the liver. Outer layer or serosa layer, if you remove that serosa layer, you will find there is a muscular layer of longitudinal and circular muscles. Thereafter, there is a muscularis mucosa portion is there, and this mucosa portion is there. So after muscularis mucosa, you will find these are the white structure lamina propria and all the surrounded with the epithelial layer. And this is a lumen or internal cavity. So here this portion is a lumen or hollow cavity, and that cavity have this villi, and at the base of these two villi in a gap that forms a um, a space or hollow cavity-like structure that called as a crypt, and this is commonly named as a intestinal crypt. Okay, that intestinal crypt generally occurs reach up to the muscularis mucosa through which that blood vessels and lymph vessels are gone. And after that, there is a submucosa portion where duodenal glands are present. Okay, in case of this, if you take a section of a single villi, what is there? There is a lining of a epithelial tissues, and in central there is a plexus of blood vessels or plexus of vessels. That plexus comprises of lymph vessels and veins and artery. Now all together this is called as a vessels, blood capillary plexus. In that a single green color portion tube you see here, that is called as a lacteal, which is a branch of a lymphatic duct, that is called as a lacteal, present in each and every villi. Okay, then if you magnify the surface of this villi, it look like this. This is a single cell, and again that single cells have lot of microvilli. That microvilli here are projection of a plasma membrane of a respective cells. So color photograph of actual slide, if you see how you can compare this thing there, this is a single villi. Here is a single villi, and the surface is a absorptive epithelial cells. So if you more magnify this absorptive cells, you will see such microvilli on that. Okay, in middle here are all that blood vessels and lacteals are present, and this gap formed between these two villi are called as a crypt or hollow space between the villi. And all these surface around the villi, epithelial surface around that crypt, are full of goblet cells are present in that. Okay, so the, histologically in histologically in a small intestine, you will see that the epithelium of mucosa is made up of absorptive cells and the goblet cells. You will find only these two types of cells present in this epithelium of mucosa. The cells of intestinal crypt secretes intestinal juice, digestive juice, and pear patches are found in the submucosa and are collected by the lymphatic or WBC tissues. Brunner's glands in the duodenum secrete alkaline mucus.